Around the bacterial metabolism of methane in a number of different environments, and also some more recent work that we've been doing in the marine environment, and that involves the metabolism of methanol. And again, I'll try, well, I think on the next slide I have the sort of outline, so I'll tell you about some of our ongoing work and just a broad overview of the research going on in my group. Then I'll take you through a bit of information about the principal organisms that we work with, the methane oxidizing bacteria, their metabolism and the key enzymes that they use to metabolize methane. Then a bit about the ecology, using a couple of rather more exotic environments that we've been sampling. Tell you a bit about a technique that we've been using to track the activity of these organisms in the environment, and then finally combine the two together to tell you a bit about the metagenomics work that we've been doing and how we're actually using this to focus down on specific types of metabolism in the environment. So just sorry, this is a little bit wordy, but just a brief overview of the research in my laboratory. We've been working on methane oxidizing bacteria, the metabolism, the physiology, biochemistry and molecular biology of methane oxidizing bacteria for many years. And more recently moving into the ecology and the environmental aspects of these organisms. So again, we have a number of representatives in culture, and I'll come back to these in a second. So we have a number of genomes available, and as I say, we're doing metagenomics as well. We've been looking at a variety of different environments, looking at methane cycling and chemoautotrophy. I'm just going to highlight three areas: the oceans, actually it's coastal water rather than the oceans in this particular case, an unusual cave system, and very, very briefly an unusual lake system. So we don't depart too far from the one carbon molecule. Again, methylamine and methanol are also what we call one carbon compounds, or methyllotrophic substrates. And again, I shall be talking about this volatile oxygenated compound, methanol, with respect to the marine environment. I'm not going to talk at all today, but a more recent project has been looking at isoprene metabolism. Isoprene is probably the most abundant trace gas after methane in the atmosphere, produced in huge amounts by trees. And again, if anybody's interested, I can have a chat with them afterwards. We're also looking at other gases like dimethyl sulfide, which is one of the most abundant organic sulfur gases, and again, work on the sea surface microlayer and C1 metabolism in the rhizosphere, so plant-associated methylotrophs. As I say, what I'm going to do today is to concentrate on using some examples from our work with methane oxidizing bacteria and then looking at methanol metabolism. So this slide really summarizes probably about 25 years of work in our laboratory. And again, I've highlighted the methane oxidation pathway. To some extent, it looks like a rather simple, straightforward pathway, but as with many of these things, it's not quite so straightforward. But methane is oxidized to methanol, and it's this step that makes these organisms unique. And of course, they're of interest because methane is one of the most stable and most simple organic molecules, and it actually takes quite a lot of energy to activate it and convert it to methanol. And I'll come back to that point in a second. So the methane can be oxidized to methanol, and again, there are many organisms that also oxidize and grow on methanol but don't grow on methane, and these are sometimes distinguished from the methanotrophs. They're called the methylotrophs. There are other related compounds like methyl halides, methane sulfonates, or sulfur derivatives of these C1 compounds, and as I mentioned just a second ago, dimethyl sulfide. And all of these feed into the central carbon metabolism of these methylotrophs. And then there are specialized pathways, which I'll come back toward at the end, by which they actually convert those one carbon compounds into cellular constituents. And there are specific methylotrophic carbon assimilation pathways. Some of the intermediates, formaldehyde or carbon at the oxidation level of formaldehyde, is actually dissimilated to carbon dioxide, and it's these steps that the organisms use to generate reducing power and energy for biosynthesis. And some of the bugs actually fix the carbon dioxide, although the majority of them actually just vent that to the atmosphere. And our approach really is to understand the biochemistry, the physiology and biochemistry of these key steps, and then look at the ecology of these organisms targeting specific steps. So rather than just using 16S ribosomal RNA genes as markers, we've also developed what we call these functional gene probes, which are based on key enzymes. And again, I'll mention this in a second. Just hold tightly PMOA, MMOX, and MXAF, because these are some of the markers that we're going to use to actually find out if these organisms are present in the environment. So a little bit more about methanotrophs. Well, front end of the grant application, I suppose. Why study them? They have a unique physiology. They grow on methane as their sole carbon and energy source. Largely, these organisms don't grow on anything else, so they're specialist organisms. What I'm going to talk about are the aerobic varieties here. They're present in very many environments. And molecular studies indicate that there are lots of these organisms out in the environment that we still haven't cultivated in the lab. And I'm sure you're aware of this concept of 95–99% of the organisms in the environment being unculturable or uncultivated. I prefer uncultivated rather than uncultured. It suggests they don't like opera if they're uncultured. But I don't really subscribe to that view. I think what we haven't done is we haven't spent enough time isolating these organisms. And it's the molecular information that can be derived from these studies that I'm going to tell you about that will enable us to actually isolate these sorts of organisms. Okay, so the methane oxidizers are really important because they mop up a whole load of that methane that's produced by methanogenic archaea before it actually gets released to the atmosphere. So they contribute hugely to the global methane cycle. In terms of there's a renewed interest in methane oxidizing bacteria in biotechnology, and certainly it's a relatively cheap substrate from which you can make chemicals, and some of the enzymes that I'll mention only briefly today are actually metabolically or enzymatically very versatile, and they can not only oxidize methane but many other compounds. And there's a list of perhaps 150 different alkanes, alkenes and aromatic compounds which these types of oxygenases can attack. And so there's been interest in the past in degradation of environmental pollutants such as trichloroethylene or trichloroethene. And the key enzyme here is the soluble methane monooxygenase, which I'll come back to talk about in a second. So a lot of interest in production of both low and high value compounds using methanotrophs. So this just gives an overview, and I'll flesh it out a little, but the methane oxidation pathway you've already seen. The principal enzyme there is methane monooxygenase, and this comes in two different varieties, a membrane-bound enzyme and a soluble cytoplasmic enzyme. More about those in a second. I've talked a little bit about the pathways, and I've given you a fairly simplified diagram of the pathways. They use these rather unusual cycles to assimilate the C1 compounds into cellular biomass. And they're rather unusual. So this is an electron micrograph of one of the methane oxidizing bacteria, and I don't know if you can see clearly, but there are membranes arranged around the periphery of the cell. Obviously this is rather unusual for bacteria, and these are believed, although it's still not absolutely understood, but it's believed that these are the sites of one of the methane oxidation enzymes. And so you can have, again, I won't go into the detail or the taxonomy, you can have type 1 or gamma proteobacterial methanotrophs and type 2 or alpha proteobacterial methanotrophs. We have probably about 20 different genera now, and again, I've just highlighted a couple. Methylococcus capsulatus strain Bath, which was isolated from the hot springs in Bath, has been a workhorse organism for the biochemistry and molecular biology. Methylocinus and Methyloceller is another organism that we've been working with in some detail. So I guess the bottom line here is there's quite a lot known about the physiology and biochemistry of the methane oxidation process, and we've got a fairly good handle on the key enzymes that carry out these particular processes. The soluble methane monooxygenase is a soluble diion center monooxygenase. This is the metabolically more or catalytically more versatile of the two enzymes. And not only does it oxidize methane and convert it to methanol, but it will also co-oxidize things like alkenes. So for example, it'll take propylene and convert it to propylene oxide. And it will even take quite large molecules, naphthalene, two benzene rings stuck together, and it will actually stick an oxygen on the benzene ring in a couple of different places. So that's rather unusual for an enzyme, of course, because we tend to think of the exquisite specificity of enzymes, and this is a very, very versatile enzyme. So again, that's the one that's been interesting in terms of the biotransformations and biocatalysis. This is found in a number of different methanotrophs, and again, this is, I try not to give you too many phylogenetic trees, but this is just a 16S phylogenetic tree of a number of representative methane oxidizing bacteria. And really, it's only there to remind me to tell you that we can find these organisms in a variety of different environments. So for example, the red ones, they're found in moderately thermophilic environments, Methylococcus and Methylocaldum. We have psychrophilic or psychrotolerant representatives in blue there. The green guys actually grow at very high pHs, up to about pH 11. And then the Methylocella palustris is a rather unusual methanotroph. Again, we've been working with in some detail. And that's one of the few that actually grows on multi-carbon compounds as well as C1 compounds. There are also recently isolated thermophilic methanotrophs that grow at low pH as well. So there's a lot of these guys out there, and it's actually only in the last few years that we've discovered some of these more recent groups. So again, I've really described the pathway. This is the key step that makes these organisms unique, catalyzed by the particulate methane monooxygenase, and then a methanol dehydrogenase, which I'll come back to later, converts the methanol to formaldehyde, and then that carbon is assimilated into the cell or dissimilated to CO2 to generate reducing power for biosynthesis. So in many respects, a relatively simple metabolism, on the surface at least. There's a lot known about the biochemistry, and this is largely due to my former PhD supervisor and close colleague at Warwick, who sadly passed away a few years, Howard Dalton. He really pioneered a lot of the work on the particulate methane monooxygenase and the soluble methane monooxygenase. And again, I won't go into too much detail, but the soluble MMO, soluble methane monooxygenase, this is the one with a wide substrate specificity, and the particulate methane monooxygenase is the enzyme that is actually prevalent in virtually all of the methanotrophs. It has we showed a long time ago now that this has a high degree of identity with an enzyme from chemoautotrophs, ammonia monooxygenase, and these organisms are specialists at using ammonia as an energy source. So we know a lot about the biochemistry of these enzymes. Again, through Amy Rosenzweig's work in Chicago, she's actually looked at the X-ray crystal structure of the particulate methane monooxygenase, and we know that this is a copper-containing enzyme. So again, I've told you about the soluble methane monooxygenase. This is the one that's been of more interest in terms of the biocatalysis and bioremediation. And again, there's an X-ray crystal structure. This just shows the hydroxylase component, so it's a multi-component enzyme system. And again, I don't know if you can see very clearly, within the alpha subunit of the hydroxylase resides this diion center, and this is the site of catalysis. So this is the business end, if you like, of the enzyme system. And it's the gene encoding this particular polypeptide that we're using as a marker, because we know from all the biochemical work that this is actually encoding the active site of the soluble methane monooxygenase. And again, another story for another time perhaps, but we've been doing site-directed mutagenesis on the soluble methane monooxygenase to alter its catalytic utility. So to summarize the sort of biochemistry and molecular biology, the soluble methane monooxygenase is only present in some methanotrophs. It has a broad substrate specificity. It's an iron-containing enzyme. and we know that it's expressed under certain growth conditions, under low copper to biomass growth conditions. These are the enzymes, these are the genes encoding the polypeptides that make up the soluble methane monooxygenase, and again, we know quite a lot about the regulation of this enzyme system as well. The particulate methane monooxygenase is a copper-containing enzyme, and this is expressed when there's plenty of copper available for the cell. Again, we know what the genes are, and I've just highlighted two, MMOX and PMOA, because these are the two genes that we're going to use as if what we call, it's not a very good term, but functional gene markers to look for the presence of these types of organisms in the environment. Again, I won't go through the regulation, I think I'll skip that, but moving on to the ecology and environmental aspects of these organisms, our knowledge of the metabolism of these bugs has allowed us to actually develop a number of different molecular tools to look at the distribution, diversity, and activity of these groups of organisms in the environment, basically to find out who's there and what they're doing. So if we look at the molecular ecology, well, we have a large sequence database of 16S ribosomal RNA genes, these phylogenetic probes, and again, this has allowed people to build a taxonomic framework for these organisms, and also to use these organisms in fluorescent in situ hybridization to target specific groups. So we can use the 16S ribosomal RNA retrieval from the environment to see what's there. But we also have what we call these functional gene probes, and again, MMOX has been used for looking to look into the environment to see if methanotrophs that have the soluble methane monooxygenase are present, and the PMOA has been used because it's diagnostic for nearly all of the methanotrophs that we have in culture in the lab. And then MXAF is another gene marker that I'll come back to, methanol dehydrogenase, which catalyzes the second step in methane oxidation and also catalyzes the first step in the metabolism of methanol by other methyllotrophic organisms. So we have a large database of these sequences. I'll just give you a brief example with MMOX. So this is what we used to do a number of years ago. We'd extract DNA from the environment. We'd use PCR to amplify MMOX genes. We'd clone these and sequence these. And obviously in the early days we used to use small clone libraries. Now we can do amplicon sequencing and sequence thousands of these from any particular environment that we care to choose. But when we compare these sequences, MMOX sequences from the environment with those from extant methanotrophs, we can actually get a fairly good idea of who's there in the environment. But of course this doesn't necessarily tell us if they're active or not. But I'll give you one example. So this is I don't know if anybody has seen this, it's a crater lake in India called Lonar Lake, and it's a lake which is actually around about pH 9.5, so it's quite an alkaline lake due to the basalt there, and there's a lot of methane oxidation activity in the surface sediments. So this was an ideal place to actually test some of our molecular ecology techniques. And again, the bottom line is we retrieved MMOX sequences from that particular environment, and we compared them with the MMOX sequences from extant organisms, and you can see that the majority of them were very closely related to the MMOX from Methylomonas species. And so this tells us that this is the major dominant type of organism present, and these sorts of organisms have been isolated subsequently from this environment. So there are some interesting outliers as well, and again, I haven't got time to go through this, but this is one way of gene mining, if you like, and actually generating new enzymes. And of course, we take this sequence, which we can tell from the signature amino acyl residues that it's a methane monooxygenase, but it's quite different to some of the others. So we do have the capacity to be able to clone this into an expression vector and look to see if there are autocatalytic properties. But I'm here to talk about the ecology, so back to the job in hand. The question we then have to ask is, are these sequences from the most active bacteria in that environment? Because these organisms, like many others, form resting stages, and in these sediments, they could actually just be sitting around. The DNA's going to remain there. And when we collect that, that will be reflected in clone libraries. So that was the question that we and many others about 15 years ago were starting to ask. Are the sequences that we're retrieving from organisms that are actually the most active, or are they just sitting around there doing nothing? And to do that, we developed a technique known as stable isotope probing. And the principle is this is very, very simple. If you grow a methylotrope with a C13 labeled substrate, for example, methane or methanol, and you can buy 99% plus enriched C13 methanol and methane. So if you feed that to an environmental sample, only the bacteria, or only the microorganisms, I should say, that actually grow, are actively growing on that compound, will take that C13 into their cellular constituents. And of course, what we rely on then is cesium chloride gradients to actually separate out. So if we take some soil and we feed it with C13 methane or methanol, extract DNA directly from that environmental sample, run it on a cesium chloride buoyant density gradient, we can actually collect the heavy DNA and separate that heavy DNA from all the other bugs that aren't actually assimilating that C1 compound. And if we can do this with environmental samples, it should reveal which methylotrophs and methanotrophs are the active ones. And so just to summarize the sort of technique that we use, it's a very simple setup, and I'll show you another example in a second. Once we've got that heavy DNA, which represents the genes and the genomes of all of the bacteria that are actually metabolizing that target substrate, we can obviously do straightforward cloning and sequencing, 16S ribosomal RNA technology. We can actually challenge it with microarrays. We can actually clone. We can do metagenomics. You can also collect the protein at the same time, and you can also collect the cell wall components. And so there are a variety of different flavors of stable isotope probing. So you can collect the RNA and look at it. And again, I'll come back and talk right at the end of a little bit of work we've been doing with protein stable isotope probing as well. But this is the general principle. So in theory, the organisms that are active in that environmental sample should allow us to the possibilities to capture their DNA or RNA and then interrogate that C13 labeled RNA. So I'll give you an example from a rather unusual cave system. Again, this is probably a whole seminar in itself, but it's an interesting example. And this is Movila Cave in Romania. It's adjacent to the Black Sea, and it's a completely enclosed ecosystem. And the whole ecosystem, and I'll show you the dynamics of the ecosystem in a second, is actually driven by methane and probably also by chemoautotrophy. So fairly close to the coast, and again I'll show you on a map in a second, you've actually got a cave system, and you can actually come down a shaft here and crawl along here and get to what's known as the lake room. There's a little bit of a lake there. There's this underground lake which is actually being fed from thermal waters deep below the surface. It's got lots of methane in it. It's also got hydrogen sulfide. And within these air bells, particularly the second air bell, I don't know if you can see it, this is a photograph, but you can see this microbial mat on the surface of the air bell. And so what we thought at the time was that this microbial mat was actually consisting of organisms that would be thriving on the methane and carbon dioxide and other chemoautotrophic substrates. So this was an ideal system with which to test our DNA stable isotope probing. The reason we think that the ecosystem is actually driven by methane, in this second air bell, there are rather unusual invertebrates. And again, I won't go through I'll just show you one picture. So this is, just to orientate you, this is Romania, this is the Black Sea, this is the town of Mangalia. And I don't know if you can see this little depression here, it's blown up on Google Maps. But what's happened, the limestone karst has dropped hundreds of thousands, probably even millions of years ago, and it's actually created this cave system. And there may well be many other caves here. It's not a rather unassuming entrance to the cave, it's just a concrete block with a manhole cover on it. And this is a highly protected environment, and the reason being is that there's no naturally known entrance. This was discovered when they were looking for sources of hydrothermal energy. There's no light, so there's no chemosynthesis. There's no photosynthesis, so any life in there must actually be driven by chemosynthetic reactions. There's no fecal streptococci, and there's no isotopes from the Chernobyl disaster, which again indicate that this has been completely isolated until it was broken into in 1985 by this drilling. As you can see, it's a very small entrance. You go down on 20 meters on a rope, and what we did there was actually analyze the whole ecosystem. But what we're really focusing on at the moment is the chemoautotrophy and methanotrophy. So you've got methane, you've got carbon dioxide, and other energy sources such as ammonia and hydrogen sulfide. And this is what makes this cave unique. Within the cave system, you've got a whole variety of invertebrates which appear to be feeding off that microbial biomass. So these microbes are the start of the food chain within that cave ecosystem. So there's about 50 species which are endemic, and quite a lot of them have become troglomorphic. They've got no eyes, and they've developed into they've developed with longer appendages, presumably to find their way around in the dark. But just focusing for a second on the first stages, we thought this would be a great place to actually test out our DNA stable isotope probing, because the evidence was that there were methanotrophs in these microbial mats, and wouldn't it be fun to actually start to follow the carbon flow through this system. We haven't got a great deal further than the microbial system at the moment, but I think there's a lot of possibilities for the future here. So our hypothesis really was that the microbial cave food web is actually supported by bacteria that are feeding on methane and also on some of these other substrates as well. So there's us going down into the cave, and again there's just about enough room to set up these experiments. There's me in my rather fetching Guantanamo Bay suit there. So it's a rather cramped environment, but we were able to take all the equipment down that we needed and set up these experiments. But the setup is actually a very simple setup. It's just a serum vial with some mat and cave water, and then we seal it up, and then we add a small amount to the headspace. We add a small amount of methane. But this methane is C13 labeled. Okay, so what actually happens is that methane comes in. This is the target organism, obviously not drawn to scale. So the methane is actually consumed by that bacterium. The C13 goes into all its cellular constituents, and then we can isolate DNA from the mat and cave water, run it on a cesium chloride gradient, and use that C13 DNA. And that C13 DNA should be the template for all of the active methane oxidizing bacteria in that bacterial map. And all the C12, that light DNA, is from all the other bugs that are actually not metabolizing the methane. So we can do obviously lots of things with that methane in the early days, and I think because it illustrates the sort of approach that we're doing. In the early days, of course, we were making clone libraries, and we were challenging that DNA with PCR primers specific for 16S ribosomal RNA genes and these functional gene markers. Now, of course, we just do amplicon sequence, or we sequence, and in fact we are sequencing all of the DNA from such experiments. But just to give you a snapshot of the information that's going on here, so if we look at the particulate methane monooxygenase, and the red names represent the PMOA sequences from extant methanotrophs, and then all these numbers represent different clones that we've retrieved from that environment that encode PMOA. And the bottom line here is that the majority of clones that we retrieved were from type 1 methanotrophs, most closely related to methylamonas. So that's one functional gene probe. The other was the soluble methane monooxygenase, and again, if we look at sequences of MMOX that were retrieved, again, they're most closely related to type 1 methanotrophs of the genus Methylamonas. So these are telling us that Methylamonas, or a relative, is actually one of the major players in terms of the activity of this ecosystem. Again, very briefly, there are other novel sequences, so we do have the capacity for cloning these into our expression vectors and looking at altered catalytic properties. It became more interesting when we looked at the methanol dehydrogenase. So all the methane oxidizers will also have this gene because they need to convert the methanol to formaldehyde. And sure enough, we found quite a lot of MXAF sequences that encode this second step, and again they were related to methylamonas, so that worked out quite nicely. But then we got a lot of MXAF sequences that were only attributable to organisms such as Hyphomicrobium and Methylobacterium, and these are not methane oxidizing bacteria. They grow on methanol, but there's no evidence to suggest that they will grow on methane. And we have lots of these growing in the laboratory. We've never been able to grow them on methane, only methanol. And so what we think is happening is that the methane oxidizing bacteria are actually consuming the methane, and some of that methanol that's been produced in their metabolism is actually leaking out, and so these sorts of organisms are actually then starting to grow on the methanol that's been supplied by the methane oxidizing bacteria. We need to go back to this system and look in a lot more detail and actually do time course studies, but we're fairly convinced that we're seeing the start of the microbial food chain, if you like, in those experiments. So, conclusions from the Movila cave study: the methylamonas-like methanotrophs predominate and are the most active. We've isolated the methylamonas, and we've actually got its genome sequence and are analyzing it now. We think that cross-feeding of methanol to methylotrophs is occurring, and this is the start of this microbial food web. And it'll be interesting in future experiments to see how far we can track the C13 label into that microbial food chain. We've done similar sort of things with the sulfur and ammonium-based chemolithotrophy as well, and so both types of metabolism are clearly contributing to this food chain. Okay, so another thing we asked some time ago was, can we combine the DNA stable isotope probing with metagenomics? So rather than just pull out little sequences, just signature sequences, can we actually pull out, initially, can we pull out gene clusters? Remember, when we started to do this, this was in the days before high throughput sequencing was A, affordable, and B, reasonably accessible. So our approach at the time was, if you'd like, to do this focused metagenomics. We're not particularly good at bioinformatics anyway, and we thought this might be a way of actually focusing down on specific functional groups. So if we can target specific processes in the environment, we have less sequence information to wade through, and then we can ask specific questions about the metabolism of certain groups rather than trying to look at everything, which at the time was very, very difficult. And in fact, we achieved this, and we sort of put this aside for a little while because, again, I won't go through the detail here, but you can probably recognize this gene cluster which contains PMOA. This gene cluster was retrieved from a back library, so a large library which had actually been generated using C13 DNA from a forest soil. And again, to cut the story short, we actually isolated, we'd already found the PMOA there, and then we started to look for bigger chunks of DNA. And this was back in 2006, just around about the time it was actually starting to become more fashionable and less expensive to sequence large amounts of DNA. So this was a sort of proof of principle experiment, but there are associated problems with this because we use very high amounts of methane and very long incubation times, and of course the longer the incubation time, the more likely you're getting cross-feeding of that C13 label to other organisms. So we moved to the marine environment, and again I'm just going to describe in about 10 minutes some work that we've been doing on a Gordon and Betty Moore funded project under their marine microbiology initiative. I think we were very lucky. I think we're the only one of very few groups outside of the UK to actually get funding from Gordon and Betty Moore under this initiative. And really, our mission there was to do this focused metagenomics with marine methylotrophic bacteria, combining the DNA stable isotope probing with metagenomics. So what we're doing is we're using the methylotrophs as a model system to look at communities and also to develop single-cell ecophysiological methods using Raman microscopy. And again, if if anybody's particularly interested, I can talk to them about that later. And really to investigate how important these aerobic methylotrophs are in carbon and nitrogen cycling in the environment. And again, I'll acknowledge the people who are doing this. The majority of the work I'm going to describe now is by Carolina and Martin, supported by Alex Howard and Ollie Burns. And again, we have collaborators who are helping us get the seawater with the Rahmen fish, and again, supplying seawater. And then Martin von Bergen and colleagues in Leipzig. Martin Taubert actually worked for his PhD project with this particular group, and so what we've actually done is we've actually started to combine some protein stable isotope probing and metaproteomics, and I'll just try and tie the two together at the end. So why the marine environment, and why methanol? Well, methanol is actually produced in very large amounts. Again, I can't remember the numbers now, but it's actually produced by plants, so there's quite a good terrestrial cycle which is well documented. In the marine environment, it's actually probably produced from algae and phytoplankton, maybe degradation of bacterial material, and obviously in coastal environments probably there's a contribution from the land as well. But all we need to know for this is that the production and consumption terms are very, very uncertain in the global methanol cycle. But we know, of course, there are organisms, I've told you already, there are plenty of bugs out there that can consume methanol. So let's just jump to, again, some figures just to show you the uncertainties. If we look at the estimates of global methanol production and consumption in the oceans, you've got a couple of numbers here. I think this is in teragrams per year. From 0 to 80, the sources are less than 0.1, and then there are other numbers here which are clearly it's just almost random number generation. The bottom line is that the global budget in the marine environment is extremely uncertain, and probably it's not that much better in the terrestrial environment as well. There have been fairly recently some estimates of methanol concentrations in seawater, and again they range from 20 to 500 nanomolar, depending on which part of the marine environment that you're looking at. Again, these figures are somewhat misleading because you're looking at a standing concentration, but we do know from measurements that there's quite a lot of methanol around. And there's quite a lot of methanol in rainwater as well, 10 micromolar, which was a surprise to me. Okay, so this is our study site. It's just off the coast of Plymouth, station L4. A lot of long-term environmental data there associated with this station, and it's relatively easy to access. There are also methanol uptake rate measurements there as well. So just to remind you, we've got these molecular probes. We've got 16S ribosomal RNA probes, and again we've got these functional gene probes. We've got this large database of sequences. So if we retrieve sequences, we can actually match up with the organisms that we've got growing in the laboratory. So these are cultivation-independent approaches with marine methylotrophs. Our initial forays into the marine environment at L4 quite clearly showed, and again I won't go through the detail, retrieving MXAF sequences from that environment and 16S ribosomal RNA sequences. It was quite clear that there was an organism, a documented methylotroph called Methylophaga, that was actually dominating this environment. So there's potentially a novel group of marine methylotrophs present there, and we showed that a number of years ago. With the seawater experiments, it's quite nice in a way because it's not as, shouldn't say as complicated as soil. It's cleaner than soil, and again there's low bacterial biomass, which I'll come back to in a second, but we knew that microorganisms would respond quickly. If we just take seawater and put a little bit of methanol and nutrient in, it goes cloudy, so we know these bugs are active. And again, we use much lower substrate concentrations in these experiments, down to 1 micromolar methanol over much shorter time periods. And again, I won't go through the detail, but in terms of the metagenomics, because we're using small amounts of C13 labeled substrate, that actually, and short incubation times, that alleviates the problems of cross-feeding. But what it doesn't allow is capture of that C13 DNA in sufficient quantities to be able to do metagenomics. So these particular experiments were actually geared up to ask the question, can we use whole genome amplification techniques using multiple displacement amplification to take that C13 DNA, bulk it up, and then analyze it in detail? And again, the bottom line, this is a relatively old experiment, but the bottom line was that making phosmid libraries with that DNA, we could actually, we didn't have enough money to sequence the whole lot at the time, but using targeted assays, we could pull out gene clusters which encoded MXAF, there's the target gene, and all of these other MXAF genes are actually accessory genes involved in methanol oxidation. And this MXAF was actually very, very closely related to the methylophaga that we'd retrieved in the previous experiments. So this was again a first proof of principle. But I just want to tell you very briefly about the current experiments that we're doing, because we've been able to, so we do C13 incubations with the seawater and methanol in the presence of methanol. We do C12 controls, and that's important if anybody's interested in the technical detail. I can discuss that with them afterwards. We incubate and then we extract the DNA and separate it out, and we use what's now a very old-fashioned or perhaps unfashionable technique of denaturing gradient gel electrophoresis to actually show that we've actually enriched specific sequences. So this is just a 16S profiling technique. So this just tells us that the SIP experiment has worked okay under those particular conditions. So we have a number of options then. We can actually take our PCR primers and look at MXAF genes, for example. We can take 16S PCR primers and look at the taxonomy of all the organisms that have been labeled with the C13 methanol. And if we want to do metagenomics, we can take some of that DNA and bulk it up using 529 polymerase and sequence it. So just a snapshot of the data here. Actually, this is probably just reiterating what I've already said, but we did time courses over short periods, 3, 6, and 10 days, and followed the uptake of methanol. So again, we look at consumption of methanol using gas chromatography. So we know the stuff is being consumed. And then in the case of the metagenome analysis, we amplified the C13 DNA using 529 polymerase. And we also combined this with protein stable isotope probing. So if I just go back to this particular stage, you can actually, once you've collected the cell material, you can also collect some of the protein and purify away the protein from all the other gunk in there and actually analyze that as well. Okay, so what did we find? If we simply looked at the distribution of 16S ribosomal RNA genes in the heavy DNA, this is T0, this is the unfractionated, and this is the fractionated. And this is looking at the different classes of 16S ribosomal RNA genes. And again, to cut a long short story short, we have a very high enrichment of organisms in the gamma proteobacteria, and when we focus down on the specific genus, it turns out that these are methylophaga. So we've got that enriched population after 3 days with low concentrations of seawater. So we've got methylophaga enriched. So that's the first piece of evidence. If we look at the MXAF genes, again, sorry, you probably can't see this, but again, the majority of them are most closely related to methylophaga. And there's an alternative methanol dehydrogenase that's present. So again, this confirmed that methylophaga-like methylotrophs are present and enriched in this marine environment. And these are the active ones that respond to that input of methanol. So with the metagenome, so we've taken that C13 DNA, that small amount, we've amplified it. Again, this just shows 2 duplicates, and this just shows the raw reads that have been assigned to different taxonomic groups. And again, you can see that the bulk of the sequences come from methylophaga. When we actually look at this more closely, because this is a highly enriched set of DNA sequences, you can actually assemble the metagenome. And what I'm just going to tell you about briefly is the genome that we've been able to assemble from this methylophaga directly from the environment without having to cultivate it in the laboratory or purify it in the laboratory. So it turns out we can actually get 8 contigs, and the nearest known relative is Methylophagous thiooxidans that was isolated by a colleague a number of years ago at Warwick. So this just shows the genome from the extant Methylophagous thiooxidans, and this just shows us the 8 contigs that are actually comprising our what we call the L4 genome, but it's this new Methylophagous species. And again, by comparing it with the known genome sequence, it looks like we've got about 99% of the genome we've captured using this method. So again, although this is an enrichment, this I think is quite a nice proof of principle that we can actually, using the DNA stable isotope probing in conjunction with the metagenomics, we can actually capture the genome sequence of an uncultivated organism without having to try and isolate it in the laboratory. We think we've actually isolated it as well, but it's an interesting proof of principle. So very briefly, you can interrogate that genome, and again, I'm not going to bore you with all the detail, but again, if we look at all the genes involved in methanol metabolism, again, you've seen a similar thing earlier. All of these genes are involved in methanol metabolism. That's indicated by MXA. Here is F, the one that we've been using as a molecular marker. And the metagenome sequence of the Methylophaga matches up very closely, almost, well, not quite identical, but very, very similar to the Methylophaga thiooxidans that was isolated before. So that's the methanol metabolism. Again, I won't bore you with the detail here, but there are accessory genes and other genes involved in methanol metabolism, and we can analyze these in detail. And again, they look very, very similar to that cultivated Methylophaga. We're also looking at methylamine metabolism in the marine environment, and these Methylophaga, the majority of them also grow on methylamine. And again, we've got the capacity, the metabolic blueprint is there in this metagenomic sequence for these organisms to grow on methylamine. And again, I won't dwell on the detail. If we look at carbon assimilation pathways, let me just remind you that the methylotrophs have these rather specialized carbon assimilation pathways. So in addition to glycolysis, Entner-Dudroff pathway, pentose phosphate pathway, and the TCA cycle, the metagenome shows that 2 of these are complete and 2 are incomplete, and this is compatible with what we know about the physiology and biochemistry of these types of methylotrophs. Again, if we look at the ribulose monophosphate cycle, this is complete, and we can actually even look at which variants of that pathway it uses. It doesn't have, as we would predict, the serine pathway or CO2 fixation. And again, we can look at ammonia assimilation. So the bottom line is we can build up a metabolic map from the metagenome sequence of that Methylophaga that's responding to additions of methanol. And so all of these pathways, these central pathways for carbon assimilation and ammonia assimilation are present. And actually the TCA cycle is not complete, and that's what we would predict. So again, this is a metabolic blueprint, so we're just looking at DNA. So are these genes actually expressed? Well, we could do metatranscriptomics, but what we were able to do through our collaborators in Leipzig was actually to look at the protein. You remember that we'd isolated from that original SIP experiment, because the protein will actually, well, I suppose those proteins could be inactive, but if the proteins are there, it gives much better evidence that that type of metabolism is actually occurring. So the green lines indicate where we were able to find peptides matching up to the enzymes in that protein SIP experiment from exactly the same pot that we got that metagenome sequence information out. And just to run back a second, you can see that virtually all of the enzymes that we look for and all the pathways are present. Here's methanol and its dissimilation to CO2, assimilation, all this carbon shuffling, etc. Again, all of the green arrows indicate that those proteins are present. The ones that aren't are this variation of the ribulose monophosphate pathway, and also methylamine dehydrogenase isn't present. And that sort of makes sense, and this probably also makes sense because we didn't feed them with methylamine. We only fed them with methanol. So methylamine dehydrogenase is actually only induced when methylamine is present. So that again matches up quite nicely. So the bottom line is the protein CYP data verify the data, the information that we've got from the metagenome regarding the likely metabolic pathways that this uncultivated methylophaga is actually using in the CYP incubations. So again, just one final thing, and again, we're only just starting to realize the implications of this, and I think I would need to consult the protein CYP people in Leipzig to give you a much better indication of exactly what's happening here, but they assure me that by looking at the C13 incorporation into the proteins that they get in these protein CYP experiments, they can actually distinguish between direct uptake of C13 into substrate and then indirect or cross-feeding on C13 metabolites. And we're still trying to delve into the data, but again, perhaps I shouldn't dwell on it, but the bottom line is if we actually look at the distribution of the peptides that are labeled with C13, we can actually see the components, those peptides that are identified that are actually from methylotrophs and those that are from non-methylotrophs, so presumably this is indicating cross-feeding. And if we look at the difference between, for example, 3 and 10 days of incubation, you can see that this labeling of peptides shifts. So presumably, if we do that carefully enough and follow time courses, we'd also be able to see the shift in the labeled peptides that are from the cross-feeders rather than the primary consumers. And again, if we couple this with the metagenomics, that should actually give us some nice indication of flow of carbon through the ecosystem. So we can use that sort of approach to go back to Movila Cave. Okay, so to summarize the L4 CYP experiments, we've got enrichment of an uncultivated methylophaga. We've managed to retrieve nearly all of the genome from that organism, constructed, reconstructed metabolic pathways, and verified them by the metaproteomics. This organism also got the potential to grow methylamine. We've actually done the methylamine CYP experiments as well. Now we're analyzing the data. So this is really proof of concept that you can actually combine the DNA and protein stable isotope probing to actually start to get more information on uncultivated organisms that are key players in the environment. And so the last slide, the penultimate slide, really, I hope I've convinced you that this type of approach, this focused metagenomics, enables you to focus down on specific metabolic groups. Of course, the DNA stable isotope probing is only one of a number of tools in our sort of molecular ecology toolbox, and it needs to be carried out in conjunction with a whole variety of other techniques as well. And really, I suppose the bottom line, and I mentioned this before, we can then use all of that information to go back into the environment and cultivate those organisms which are so-called uncultured or uncultivated. And I think if we try hard enough, we'll get these sort of organisms into the laboratory, and we can actually look at environmental regulation in the laboratory, then go back into the environment. So just to acknowledge the people who've done the majority of the work here, Alex Howard, Carolina Grobe, Martin Taubert, and Ollie Burns have done most of the marine work, and Deepak was involved in the Movila Cave and the Lona Lake work. And again, I should just acknowledge the funding sources on the bottom. Thank you very much. And I'm more than happy to take questions after I've had a drink.
Well, I would have a couple of questions. One is, what do you know about like the seasonal development of these methanotropes? For instance, at station L4, where you have a very strong seasonal cycle. Yes.
Well, interestingly, we try and coincide our sampling with the phytoplankton blooms, because there's more biomass there, and it's quite clear that there's a cycle, and so the amount of methanol that's released or so I should say, the amount of methanol that's been measured is higher either during or at the end of these blooms. Although interestingly, we did some sampling back in September, and actually the experiments didn't work for various reasons, and we went back a couple of weeks ago and we're still getting very, very high activity of these sorts of organisms. But clearly, yes, there's a seasonal cycle, and there could well even be a diurnal cycle, but we haven't got the technology to be able to focus down to that with these sort of techniques.
A completely different question that I had is, you showed that for the soluble, what was it? Methanol monooxidase.
Methane monooxygenase, yeah.
You needed a di-iron center.
Yes.
Large parts of the oceans where iron is limiting. Has anybody looked like how the methanotrophs deal with that problem?
It's interesting. When we look at methanotrophs in the marine environment, again, we have another really interesting site which we're looking at methane oxidation, and it's off the island of Elba. About 200 meters, there are these shallow methane seeps. So they're very, very similar to the deep sea hydrothermal vents which are gushing up methane, but they're far more accessible. And we've been doing methane SIP experiments there. We're in the middle of analyzing those data at the moment, but it's quite clear from those experiments and experiments we've done over the last 20 years that methanotrophs that have soluble methane monooxygenase are not very prevalent in the marine environment. So whether that's a total coincidence or not, I don't know. In the old days when we just had our PCR assays, we actually did a transect of the tine going from less salty out through the estuary, river estuary out into the sea, salty end. Particulate methane monooxygenase primers generated sequences, signals all the way out into the salty stuff. Soluble methane monooxygenase came out into the estuary, perhaps 1.5% salt, and disappeared. Circumstantial evidence that maybe the iron is limiting, I don't know. I really don't know. But that seems to be the case, that the marine environment seems to have, there are very few true marine methanotrophs that have soluble MMO. I guess that's probably the best statement to make.
Is there a constant fraction of methanotroph in the bacterial population?
I'm sorry, say that again, please.
The fraction of methanotroph
Yes.
in the bacterial population, is it constant or?
No, it will vary. It will vary enormously. So if we go to landfill cover soils and peat bogs, where there's a huge amount of methane being released from that environment, the methanotrophs may constitute about 10% of the total bacterial population. If you go out into pristine soils in grasslands and things like this, they may be 0.1 or 0.01% of the population. So there's a huge gradient, and that's what you would expect, because of course that's the only thing that they can grow with the exception of 1 or 2 methanotrophs. They can only grow on methane. So if there's not methane around, they're going to be in relatively low numbers.
They're typically not more than 10%.
I wouldn't like to put a figure on it. I mean, we'll be able to get some better indication now in Elba, because these shallow methane seeps, they're only about, I think it's about 10 or 15 meters deep, and for sort of 100 square meter region of that seawater, methane is saturating. So methane isn't particularly soluble. The seawater is saturated with methane. So if you were a marine methanotroph, that's where you want to be sitting and living. So it's going to be interesting to see how many, as a fraction of the total population, are methane oxidizing bacteria. But you have to remember, they're competing for nitrogen, phosphorus, and all the other goodies. So although methane is plentiful, they still have to compete with all the other nutrients, with all the other bugs.
The lack of specificity of the enzyme, the monooxygenase, do we know if these organisms contribute significantly to other cycles, like nitrogen cycling?
Well, the methane oxidizing bacteria, some of them fix nitrogen, not all of them fix nitrogen, but they won't use these monooxygenases to handle any nitrogen compounds. These are specifically for oxidation of carbon compounds.
I thought you said that they would oxidize ammonia as well.
Okay, right. Yes. Okay. So I didn't say they oxidize ammonia, but they do. So you were right anyway. So the particulate methane monooxygenase and the which is a membrane-bound copper-containing enzyme, and the ammonia monooxygenase from nitrifying bacteria, which use ammonia as an energy source, but obviously not as a source of carbon. Both of those two enzymes seem to be evolutionarily related. So there might have been some sort of progenitor organism which at one time could oxidize both methane and ammonia, and they've diverged. So one has used ammonia as an energy source, and one has used methane as a carbon source. So if you look at the Km, if you look at the affinity for methane and ammonia, off the top of my head, they're about 100-fold better for their respective substrate. So the activity of the methane monooxygenase with ammonia is about 1% rather than 100%, and vice versa. So you can get them in the laboratory, you can get them to oxidize ammonia. You give it sort of 10 millimolar ammonium and it will actually oxidize it. But out in the environment, they're not going to be able to compete with nitrifying bacteria, where they're using 100 micromolar, 10 micromolar even. Yeah. So no contribution to nitrogen. No contribution to nitrification, but probably in environments where there's a lot of methane, a significant contribution to nitrogen fixation. And peat bogs, where there's a lot of methane, not much nitrogen, less nitrogen, you find nitrogen fixing methanotrophs. George.
Going back to the iron question, either from the genomics or from physiological experiments, are these guys prolific producers of siderophores?
It's an interesting question. I don't know if you, George and I go back quite a while. I don't know if you remember, George, when we were at Warwick and we had the big collection of methane oxidizing bacteria. There was one, a Methylocyanosporium, that used to look black on plates. Yeah? Okay, so we got quite excited about this because we thought it was some sort of siderophore or something like that. It turns out that if you knock out the soluble methane monooxygenase from that organism, it doesn't make that presumed siderophore. It doesn't go black. And we never went back to re-examine it. But I suspect that, that's a whole new story with a copper-containing enzyme, which requires lots of copper. There's a methanobactin, a chalcophore, that actually gets excreted into the medium and captures copper and brings it back in. I suspect under certain environmental conditions, and I bet the peat bogs would be a great place to go, there'd be quite an interesting project with iron acquisition, because they have to have it. They have to have it for other things, cytochromes, but there's that extra requirement for methane oxidation.
Just remind me about the two promoters that are controlling the particulate and the soluble. What's the impact of? They're differentially regulated with regard to copper, right?
Yes, they are. Yeah.
In an era of synthetic biology,
yeah.
What happens if you switch those around?
Yeah, good question. Good question. I suspect they're probably working reasonably independently, but I don't know. It'd be a really fascinating thing to do. I mean, we've recently knocked out the genes that we've identified in Methylocyanus trichosporium, the genes that actually produce methanobactin. And again, there's some interesting but rather difficult to explain regulatory phenomena that we're seeing there. So we can discuss it later.
Yes,
Please.
the other question, the old question is, formaldehyde's a key intermediate there. How do you think they handle that? It's pretty toxic. Is it just that it's getting assimilated so rapidly?
I think it's assimilated so rapidly, so there are at least 4 different mechanisms that nearly all of them have to be able to capture that formaldehyde immediately. I don't know if you noticed on one of the one of the early slides when I was just talking about the overview, I mentioned isoprene. So there's an interesting link between the methane, because the isoprene degrading bacteria actually make an epoxide, and of course that's also quite toxic within the cells, highly reactive, and they conjugate that immediately with glutathione. So they've got mechanisms by which they can deal with this toxic intermediate, and I think the methanotropes are really fine-tuned to do that. So there's a membrane-bound formaldehyde dehydrogenase, there's a soluble formaldehyde dehydrogenase, and then the assimilatory enzymes as well, which I'm told have very, very high affinities. It's probably just sucking it up as quickly as it can get produced.
So can you convert a methylotrope into a methanotrope just by moving the MMO cluster?
A lot of people are, in the era of synthetic biology and systems biology, a lot of people are talking about doing it, but we've had a lot of experience with attempting to express the soluble methane monooxygenase in heterologous hosts, and we've been doing site-directed mutagenesis, but we use a homologous expression system. And I always use the analogy with nitrogenase and nitrogen fixation. You have 3 peptides that make up the enzyme, that's easy, but then you've got about 15 accessory genes encoding proteins that actually put the molybdenum cofactor into the nitrogenase, the iron actually help assemble, and I think it's an assembly issue. That's my hunch, that it's going to be rather difficult in a heterologous host. So methylotropes don't contain those membranes, so if you're going to use the particulate methane monooxygenase, then you maybe want to move it into a nitrifier might work. lots of games to play on paper, but in practice it's actually technically very, very challenging, because we don't have a great deal of genetic systems for these organisms.
Okay, so I think it's time to move to the wine reception. Thank you very much for the interesting talk.
Pleasure, thank you.